virus. It's in the family Retroviridae and the genus lentivirus. So lentivirus might not sound as familiar, but that's the taxonomy that we're working with. All right, let's start with the structure of the virion. Like herpes virus and orthomyxovirus, it is an enveloped virus. Okay, as far as its genome, it has a segmented genome, and it's positive sense single-stranded RNA. All right, so remember orthomyxovirus were negative sense single-stranded RNA. And you can see over here at our diagram, we've got actually 2 copies of the 2 RNA strands. So segmented because it's 2 different strands. They're actually copies of each other. All right, so you would expect a virus that's single-stranded RNA to be, it's pretty straightforward typically for a positive sense single-stranded RNA virus. That can be used directly at the ribosomes to translate its own proteins to replicate itself. All it has to do is go through a negative sense single-stranded RNA intermediate. So easy, but that's not what this virus does. It uses an enzyme called reverse transcriptase. And that enzyme converts that single-stranded RNA to single-stranded DNA, and then makes the complementary copy of that, so makes it into double-stranded DNA. All right, so this was pretty surprising when this was discovered. This goes against our central dogma of the way that information is passed through the cell. There's no other life other than a few viruses that do this, that make DNA from RNA. But the advantage of this now that it has the double-stranded DNA, that enables HIV to integrate itself into the host chromosome. Right, so then it stays there basically forever in a latent state until it reactivates. So reverse transcriptase, excuse me, makes lots of errors. So basically a little bit more indiscriminately kind of throws in bases there, much more error prone than what our enzymes do when we, say, replicate our DNA or transcribe our DNA. So as a result, we see lots of new viruses that have subtle differences in their surface proteins. So, and we're going to look at those surface proteins in a minute. So throughout the course of HIV infection, these proteins are constantly changing, and our antibodies don't mount a very good response to the virions. All right, so some of those key glycoproteins on the surface of HIV. So let's see, GP120, GP for glycoprotein. That function of that is attachment to the host cell. All right, so in your diagram here, you see that over here, that's the little purple knob, and then GP41, we think that's important for fusion of the viral envelope with the host cytoplasmic membrane. And you'll find that right here. That's the little red part there. Okay, so what else you see in this diagram, don't worry about the matrix proteins or the capsule proteins. We're not going to worry about those. Here's showing you how the cell brings with it a copy of reverse transcriptase. There's actually 2 other proteins that it brings with for some reason it shows one here, the little green dots, and doesn't label it. I believe there should be another one as well. At least there's a video animation here that I've posted the link for, and I'll show you in a minute, that shows anyway another protein there. So the 3 proteins that it brings with it: reverse transcriptase, integrase, and protease. So we'll talk about those 3 enzymes shortly here. The other thing that I wanted to mention is there's 2 types of HIV, HIV-1 and HIV-2. HIV-1 is predominantly in the US and has been studied a little bit more extensively. HIV-2 shows up a little bit more commonly in Africa. All right, let's talk about transmission, and then we'll get into a little bit more detail about the life cycle of the virion. HIV is spread by direct and specific routes. So casual contact doesn't lead to infection. I can remember in the 80s when this was first coming onto the scene, people were afraid of, oh, can you get it from, sitting on a toilet seat in a public restroom or swimming in a public swimming pool? The answer is no. You can't even get it by a mosquito bite, which you think that maybe you would be, but due to the anatomy of the mosquito, that doesn't make it possible. All right, so how is it passed from person to person? We know certainly blood transfusions, and a lot of work had to be done in the 1980s to make sure our blood banks, our blood supply was safe. Sexual intercourse. Worldwide, it's mostly passed by heterosexual intercourse. In the U.S., the males more commonly get it by homosexual intercourse. Let's see, IV drug users. We're using needles, dirty needles, mother to child. About 1/3 of babies born to HIV positive mothers are HIV positive. All right, and the virus can also pass through breast milk. It's pretty wimpy virus in the environment, so it's sensitive to heat and disinfectants. Remember, those membrane-bound viruses don't survive typically very long in the environment. We've made a lot of progress in slowing this pandemic. As of 2017, 36.9 million infected worldwide. So, wow, that seems like a lot. In the US, it's about 1.1 million. Okay, the African continent has especially been devastated by HIV. The numbers that I have as of 2009, about 5% of adults in sub-Saharan Africa were living with HIV, and in some places that number was as high as 28%. So that's a lot of people infected, huge problem with tuberculosis and HIV infection at the same time. But like I said, we've made a lot of progress. In 2005, there was 5 million new cases worldwide. Now, 2018, 1.7 million new cases. So the infection rate is slowing way down. In 2005, we had 3 million deaths. In 2018, we only had 770,000 deaths. So we are making progress. As of 2011, that's the year that I have numbers for, the life expectancy for someone that's HIV positive, this might surprise you, as long as they're getting treatment, life expectancy, 70 years. Okay, we don't have a cure, but the drugs are just drastically extending lives. All right, let's look at the main points of the replication cycle of HIV. And this is the figure from your book. So pay particular attention to those surface proteins that we talked about before on the virion, as well as the 3 proteins that it carries with it: reverse transcriptase, integrase, and protease. All right, so HIV can actually utilize both a lytic and a latent replication cycle. You'll see we start out here with attachments, all right, and the little pink dots there are the GP120. That primarily is a little bit of a simplification, but it primarily attaches to the CD4 protein receptors. Remember what kind of cell that we've talked about that has positive for CD4? That's T helper cells. Now, in actuality, other kinds of cells have those CD4 receptors as well. Macrophages, dendritic cells, and B cells, as well as granulocytes, also are positive for that CD4 receptor. So HIV actually infects all these different kinds of cells. You don't really hear about that because it's T helper cells that are killed by HIV. Even though HIV infects these other cells, they just replicates in them and leaves them and doesn't really harm them. That's my understanding. All right, so it attaches to that CD4 protein. A break in the skin, a microscopic break in the appropriate mucosal membranes has allowed the virus to penetrate in. It again attaches to that receptor, and entry by endocytosis here, that's facilitated by that GP41 glycoprotein. Okay. That triggers endocytosis. At some point, once it's in the cell, the membrane, the envelope of the virion uncoats, as well as the capsid uncoats there, and then we see those 2 copies of RNA and those 3 proteins that it brings with us spilling out. Okay. First step then is that double-stranded DNA is transcribed by the reverse transcriptase. Then it's that DNA enters the nucleus where it gets integrated into the host DNA, and that enzyme, integrase, there it is a key enzyme in that process and integration into that provirus. That's the term that we have once it's integrated. Okay, so once it's integrated as a provirus, it's latent and it can hang out there for a long time. But when it reactivates, it starts transcribing more of its own messenger RNA, assembling the viral proteins necessary into a virion. This enzyme, spelled wrong here, protease function, there's some kind of cleavage that has to happen of those viral proteins to make the virion a kind of viable virion. This diagram has that cleavage happening once the virion is outside of the cell here. My understanding is that happens when the virion is still inside of the cell. But either way, that's the function of the protease. Okay, the animation that I'll refer you to has it happening inside of the cell. Okay, so this is a really excellent animation. It's just a few minutes long. If you could watch that, it has a little bit of extra information beyond what we're doing, but it has some quiz questions to go with it too, so that's an excellent resource. All right, let's talk a little bit about the drugs that we have to treat HIV. They all target the enzymes necessary for viral replication. Okay, and typically a patient is given about what we call the AIDS cocktail, so usually 3 or more drugs at once. Okay, so we have kind of 3 different big classes: drugs that interfere with replication. Okay, so these are going to be reverse transcriptase inhibitors. Okay, so let's see. Here, that's shown over here on our diagram. And more than half of the drugs that are available work this way, that target that reverse transcriptase, because obviously that's an enzyme that we don't need ourselves. Okay, some of them are nucleoside analogs. We've actually mentioned an example of this before, AZT. So a nucleoside analog, this kind of mimics one of our bases, as well as the sugar deoxyribose that goes with that. Okay, so when, say, AZT, for example, is incorporated into the viral DNA, it doesn't have that 3' hydroxyl group, and so it stops replication. Okay, some reverse transcriptase inhibitors actually bind directly to the enzyme, so they're considered non-nucleoside analogs. So again, they bind directly to reverse transcriptase in order to inhibit them. There are drugs that interfere with integration. Okay, so you guessed it, they are integrase inhibitors. And let's see, that's up here, integrase inhibition. Okay, so the viral DNA can't become part of the host genome, or that's the goal anyway. And then lastly, there are drugs that interfere with release. Okay, so these are going to inhibit that third enzyme. They are protease inhibitors. Okay, that's shown right here. Okay, so all of these drugs have side effects. They can cause nausea, vomiting, fever, tiredness, dizziness, insomnia. It's a long list of possible side effects. Usually those side effects last a few weeks when the patient first starts taking them, and then they usually go away after that. There are lots of long-term side effects as well. Again, the list is too long to even write down here, but kidney problems, liver damage, heart disease, all potential long-term side effects of taking any of these drugs. And the drugs cost about $20,000 a year. So it's quite expensive, but if a patient stops taking the drugs, the virus will begin to replicate again. So it's really important to keep up the therapy to treat HIV so that the life can be extended. And lastly, it's important to try to understand kind of what's going on in the body during an AIDS or HIV infection. So again, this is a figure from your book. This figure is tremendously complicated because we have a couple different axes. So once you kind of understand what it's showing, it's actually a pretty interesting, informative graph. Okay. So what we have here is the relative amounts of HIV in the blood and that's over here on the right axis here. That's measured by quantitating the copies of RNA in the blood plasma. This axis over on the left side is corresponding to the green line, the amount of CD4 T helper cells in the blood, and then the antibody titer against HIV. That's just the purple line. All right, so when a patient is first infected here, the HIV, rapidly replicates, and we get a big spike in HIV in the blood. It's killing the CD4 helper cells, so the CD4 count is dropping, but we're still able to mount enough of an immune response to get enough antibodies to kind of reduce the titer of the HIV in the blood. Okay. At some point, typically though, the HIV, again, it goes latent. So, this is quite a variable break in time. It can be months, it can be years until the HIV reactivates and starts replicating again. When it does, it gradually increases in the amount of HIV. Eventually, so it's killing the CD4 cells because the CD4 population is steadily declining. Remember, our B cells need T helper cell help in order to activate to become plasma cells and make antibodies. So if our numbers of CD4 are going down, then our antibody production is also going to be going down as well, until basically the virus replicates unchecked at all. A patient is considered to have AIDS once they're below this threshold of about 200 CD4 T cells per milliliter of blood. Okay, so about at this point, the patient would be considered to have full-blown AIDS. Eventually, they die not because necessarily they've got all this virus in them, but due to opportunistic infections. Okay, so all sorts of things that typically a person wouldn't have any trouble fighting off now can become life-threatening. All right, so I copied a list from the CDC of some examples of these opportunistic infections. Don't memorize these. Probably most of them are not familiar to you at all. But again, if a person has 2 or more of these opportunistic infections, then they're considered to have AIDS no matter what their CD4 count is. Okay, so either CD4 count below 200 or 2 or more of these opportunistic infections. So any of these that you might recognize, a few of these we talked about, this cytomegalovirus, remember this is HHV5. Typically, people don't have any symptoms to this except if you're HIV positive. Herpes simplex, we talked a bit about herpes simplex. Otherwise, this Kaposi sarcoma is HHV8. Remember, oops, and that's spelled wrong. But you'll see, cancers. TB is probably one of the only ones on this list that people might typically get if they have normal immunity, but much more likely to get it if you're HIV positive. And other infections like a typical person could get salmonella, but for the salmonella to go septic, that's a sign of a weakened immune system. So again, it's the opportunistic infections that eventually cause death. All right, I have one more video to share with you, and that's concerning coronavirus. So hang in there, and we'll see you next time.